Mute myself. Good morning, everyone, and thank you for joining us for the Final Outcomes press conference for the CCMI project, Increasing Coral Resilience with Assisted Evolution via Restoration. So for the past 2 years, CCMI and Dr. John Bruno from the University of North Carolina have completed a 2-year coral restoration research project in which we investigated how acroporid corals with different genetic makeup or genotypes respond to climate change-related stress. This project has been co-funded by a RESEMBID grant from the European Union. RESEMBID, funded by the European Union and implemented by Expertise France, the Development Cooperation Agency of the Government of France, supports sustainable human development efforts in 12 Caribbean overseas countries and territories, or OTCs. This project has also been supported by DART, Stuart Humphreys, the Disney Conservation Fund, Cayman Water, the All Foundation Trust, and the Ernst Kleinwort Charitable Trust. The reason CCMI began this project was because climate change and other stressors pose substantial risks to coral restoration efforts. Coral restoration is a highly valuable tool for maintaining biodiversity and supporting coral reproduction. But as the threats facing corals intensify, as shown by last summer's coral bleaching event, managers need to understand how to make their restoration projects as resilient as possible to withstand threats and harsher environments. CCMI research team built on previous work that looked at how Acropora cervicornis, or the staghorn coral, with different genes or genotypes can respond to different threats. Previous work has also found that some of these genotypes showed higher resistance to coral disease than others, and some recover better from disease infection or can survive in warmer waters than others. In this project, our team examined thermal tolerances of these genotypes, as well as other desirable traits for resilient corals, such as higher growth rates, to identify which genotypes have the highest overall resilience in order to inform coral restoration strategies. So we're now going to show a video that discusses the overall project and its findings, and what this means for resilient coral restoration practices in the future. Again, thank you for joining us, and we hope you enjoy our press conference and discussing the outcomes of this important work. In 2022, with funding from RESEMBID, CCMI and Dr. Bruno from the University of North Carolina collaborated to enhance coral reef restoration success in the face of climate change. Their goal? Understanding if different genes in Acropora corals affect their tolerance to warming waters and subsequent bleaching. The CCMI team also investigated whether the corals with increased thermal tolerance showed better growth and survival rates when transplanted back out onto the reef. For years, scientists have sought a supercoral with genetic resilience to threats like warming oceans. This research aimed to identify resilient coral genotypes, crucial for successful restoration in the face of future challenges like warming waters and coral diseases. Building on CCMI's pioneering coral restoration work since 2011, the project used corals from the coral nursery to study how specific genotypes tolerate higher ocean temperatures. Lab experiments conducted at our Little Cayman Research Center measured key traits under different thermal conditions. The 2023 bleaching event, whilst devastating, also provided real-world validation of our findings in wild-planted corals. Lab experiments revealed a genotype with the highest heat tolerance. However, some high-temperature tolerant genotypes showed lower growth rates and increased disease susceptibility. No single supercoral emerged. Instead, a mix of genotypes with different traits is crucial for overall resilience. So what does this mean for coral restoration in the future? To boost resilience, restoration practitioners should stock coral nurseries with a diverse genetic pool of coral colonies to survive the complex stressors in a changing ocean. Understanding genetic makeup is vital for future restoration success. As climate change impacts intensify, successful coral restoration becomes paramount. More climate-resilient projects offer hope for saving coral reefs by enhancing diversity, providing habitat, and aiding natural coral settlement. This buys time for these vulnerable ecosystems to adapt while global climate action and marine conservation efforts continue. So we hope you enjoyed that video describing our project. And now we're going to go right into showing you some of the results from this work. So as you heard from the video, the goal of this project was to use our knowledge about past disease history, as well as incorporate some testing on heat tolerance and look at how these differences in resilience can be used to improve our restoration practices. Next. And the reason we're focusing on the acroporid corals is because these corals have been historically impacted the most in the Caribbean than other corals taxa. So for example, you can see from this graph here that from historical times to present, we've lost a dramatic amount of the coral cover of these particular species. And this is primarily related to an outbreak of white band disease that happened in the late 1970s. More recently, coral bleaching has become the most prominent impact on these corals. And so now it's important that we're looking at both disease and temperature as threats and how we can mitigate the impact of either one of these or both. Next. And the reason is because we've lost so much of the Acropora coral cover that some of these reefs that used to be 80% covered by acroporids are now barren and do not have any or very little of the acroporids left, so much so that the acroporids are listed on the endangered species list. This led many organizations to start to instigate coral restoration practices. And some work that we have been involved in previously, for example, at Mote Marine Lab or the Coral Restoration Foundation, initiated this work, and CCMI built on their initial progress to establish our coral nursery in 2012. And we're focusing on Acropora cervicornis, or the staghorn coral, which we grow on these frames that you can see in the lower left image here. And some of our previous researchers visiting the station were able to monitor the disease outbreak that happened a few years ago. And because we know the genotypes of each of the individuals within our nursery, we can monitor how each genotype is vulnerable or resistant to that disease outbreak. And what Dr. Anya Brown found was that disease prevalence differed amongst the different genotypes, with what we call genotype G having the lowest disease prevalence and genotype K having the highest disease prevalence. So building on that, we then wanted to look at thermal tolerance of these genotypes. And so to do that, we collected small fragments of the corals from our nursery, brought them into the lab and put them in these little closed chambers that you can see here. And we monitored their oxygen production and consumption over time under different temperature regimes. And doing that, we're able to produce what we call thermal performance curves, which show things like what is the optimum temperature for performance or the maximum temperature that a coral can withstand. And these are the results from those thermal tolerance experiments. So here you can see the graphs broken up into each different genotype, which is designated by a letter. So for example, you'll remember in that disease graph, we had genotype G having the lowest prevalence of disease and genotype K having the highest prevalence of disease. So there are a few things to point out in these graphs because they're kind of confusing to look at and interpret if you don't really know what's going on here. So one of the first things we look at are things like the T-opt. So this is the optimum temperature for metabolic performance. And so we found that the highest T-opt was for genotype G, that disease-resistant genotype. And we also looked at TOPT for OB. Now, OB didn't stand out as a disease tolerant or resistant genotype, but here we can see that OB seems to have the lowest optimal temperature, which you would think might lead to it preferring colder temperatures than genotype G. However, when we look at this metric called the EH, or the deactivation energy, what we're looking here at as is the slope of this. So a steep slope would indicate that once that coral passes its thermal threshold, it's going to very rapidly decline in health. So maybe only, zero point 0.1 degree above that thermal threshold, that coral might bleach. Whereas if you have a very gradual slope, it means that the impact of increasing temperature may be less severe. So, 0.5 or 0.6 degrees higher, they still might be surviving. And so here we found differences between G and OB, where that slope is much more severe for genotype G than it is for OB, which would suggest that genotype G may be a higher threat of bleaching if that temperature threshold were surpassed. And finally, the last thing we look at is this thermal margin as well as the CT max. And so here you see, again, genotype G standing out with the lowest maximum temperature tolerance, as well as the lowest thermal safety margin. And again, this thermal safety margin is an indicator that once you pass that maximum temperature, it's very likely to have a severe impact on the coral's health. So once we did this thermal tolerance experiment, we then set up a common garden experiment. And so to do this, we set up 3 replicate dome sites. So what you see in this picture is one dome site. In this common garden, we had 3 of these, and each one had 24 corals, which represented the 8 different genotypes. And they were initially measured for total length and then assessed for health after a year. So we did this both for growth as well as health status. So our results for after a year from the health were quite interesting. And so what I want to point out here to you is that the turquoise color is disease. And what you'll notice here is that, again, genotype G, which is the second column in that graph, did not show evidence of disease. So it's persisting to show this resistance to disease. And this was what was historically the case in our initial study. Whereas, for example, genotype K, again, is showing a high prevalence of coral disease. So this suggests that corals that initially were resistant to disease are still resistant to disease, whereas those that were vulnerable are still vulnerable. So they're not gaining any benefit from having experienced that disease outbreak prior. They're still vulnerable if they were initially vulnerable. As we continued this study, the seawater temperatures continued to increase. And one of the interesting things that happened this summer was a severe bleaching event because the waters in the Cayman Islands exceeded our bleaching threshold. So we calculate bleaching thresholds by the number of weeks at which a temperature exceeds the annual average or annual maximum as a degree heating week. In the Cayman Islands, we exceeded 19.5 degree heating weeks. And for reference, the previous highest degree heating weeks was 7 degree heating weeks in 2015. So this was an exceptional heating event that occurred. We were able to capitalize on this, knowing the disease history, the bleaching tolerances from our thermal performance, as well as the individual genotypes of all of the corals that we had outplanted and have in our nursery. We were able to use that to assess bleaching by genotype again. So here, we're looking at bleaching in September, and I want to point out here OB genotype, the first one in this graph. You'll recall from our thermal tolerance data that this had the most gradual slope past its maximum temperature, which I previously suggested might say that it would take longer to bleach once it passed a thermal threshold. Well, that's what we found. In fact, we found that this coral genotype had the lowest prevalence of bleaching, and that suggests that perhaps these thermal tolerance experiments are quite valuable in predicting which corals will be able to tolerate future climate change scenarios. However, genotype G, which is towards the right of this graph here in orange, had very high bleaching prevalence. And you will note that this is the disease-resistant genotype. So here we're finding a trade-off between bleaching and disease, where those corals that were resistant to disease are actually quite vulnerable to bleaching. And when we look at that final health status data, this is shown even more profoundly. So by the time we get to September 2023, looking at our common garden experiment, you can see that a large proportion of our corals had suffered bleaching-related mortality. There's still a slight prevalence of disease, but the majority of the mortality that happened on our common garden experiment was due to bleaching. And this was genotype specific, in particular genotypes B and G bleached quite heavily, whereas genotype K showed less bleaching. And again, this was that very vulnerable genotype to disease. So it showed less bleaching, even though it showed more disease. So what we're finding overall is this trade-off between traits. So a coral may be disease resilient, but because of modifying its genotype or its phenotype to be resistant to disease, it becomes more vulnerable to bleaching. And so in summary, our data really indicate that genetic diversity is the key. We found that genotypes that showed high disease prevalence in the past continued to be vulnerable to disease now, whereas genotypes that showed low disease prevalence were those that showed genotypes that showed low disease prevalence are the same ones that showed higher thermal tolerances and bleaching resistance. So here, we're showing that there's this trade-off. So in order to maximize the potential of our restoration practices, we need to hedge our bets. We need to have genotypes of all different kinds some that are better at disease resistance, some that are better at mitigating predation, some that are better at tolerating high temperatures, so that no matter what the stress is that hits our nurseries and our outplanting communities, we have a genotype that can tolerate that. And also, I think a key point here is that the thermal performance analysis that we performed really did actually pick out who the winners and losers would be in a thermal heating event. And that's really important because this indicates that other restoration practitioners could use a similar metric to try to identify thermally resistant genotypes in their nursery. And so with that, I think we're going to open up the press conference to questions. So if anyone has a question that they would like to ask about our project, we would be happy to answer them.
We've got a question. Will CCMI be using these results in their restoration moving forward?
So the question is, will CCMI be able to use these results in our restoration practices going forward? And the answer is, of course. We have already done that. So one of the final deliverables of the RESEMBID project was creating 5 new outplanting sites. And we did that, maximizing the genetic diversity of the corals within our nursery. So every genotype that we had represented in our nursery is represented among and within each of those 5 new restoration sites. And that's the plan that we intend to use going forward.
And what practical steps can those in coral restoration take to start implementing these results?
Okay, so the question is, what practical steps can those in coral restoration take to improve their results? I think that the biggest takeaway from this is that we should not have mono or clonal restoration nurseries. We need to maximize the genetic diversity within our nursery as well as within our outplanting communities. So our disease work also showed that trees that have maximized diversity within the trees have the lowest outbreak of disease as well. And so all of these results are leading to this indication that restoration practitioners should try to maximize genetic diversity within individual trees or frames, as well as within their outplanting communities. And even if you don't know the genetic makeup of the corals, I think you can base some of this just on where you're getting those corals of opportunity from. So trying to maximize, the diversity of locations where you're collecting your fragments that populate your nursery.
Okay. So you mentioned predation. Is that something that was examined in the study? And how did it align with thermal resistant and disease resistant genotypes?
Right, so the question is about predation and whether we looked at predation and how was there an interaction of predation with thermal tolerance and disease outbreaks. And I will say that we did not actually analyze predation in particular. We noted if there were snails on our corals, but we did not incorporate that into our analysis because it was quite low rates of predation. I know that predation on corals in other locations such as Florida is quite a big issue. And luckily here, it's not as strong of an influence on our nursery as it is in other places. So we did not use that in our analysis because the level of replication was quite low. Like 1 or 2 corals had a snail on them.
We've got another one. Does CCMI have any plans for spawning these corals, which are showing higher tolerance and resilience?
Yeah, spawning work is definitely in the future, particularly because of the impacts of this last summer on our coral nursery and the genetic diversity within our nursery. This severe bleaching event definitely impacted how many genotypes we have remaining. And so going forward, we're going to try to improve the genetic standing stock by incorporating spawning and sexual reproduction with cross-fertilization.
Okay. I think, should we just check if that's answered all the questions?
Sure. If there are any other questions, please type them in. I'm happy to answer them. Or you can always reach us at CCMI at reefresearch.org and info@reefresearch.org. And we'd be happy to answer any of your questions related to this project or any of our projects at CCMI.
Should we just mention the handbook as well, and then I can post a link in the chat?
Sure. So we would like to mention that we did write a handbook outlining the results of this project. And this handbook is available online in English, Spanish, French, and Dutch. So no matter which one of those languages you speak, you can find the information as well as additional details to our methods and results outlined in that handbook.
Great. Oh, we just had one last question, actually.
Sure.
2 more. Was stony coral tissue loss disease one of the diseases specifically studied?
So the question was about stony coral tissue loss disease and if that was one of the diseases specifically studied. The answer to that is no. So stony coral tissue loss disease luckily does not impact acroporid corals. There are several vulnerable species to stony coral tissue loss disease. However, Acropora is not one of those. The disease that primarily affects Acropora is white band disease. And so that is the disease that we focused on.
And was water depth investigated in regards to the response of the corals?
So this question is about water depth and if that was investigated in response to our corals. So we actually have done previous work looking at the success of nurseries at different depths. And that work showed that that's a paper that was just published by Paul Manival. And that result was that the corals that were in deeper sand holes in about 60 feet of water or 15 meters did better than the corals that were in 30 feet of water or 8 to 10 meters. So we did, in fact, after that study, migrate all of our nurseries to that deeper site because we found higher survival rates and less disease and less bleaching the deeper we went. Our common garden experiment actually was in 70 feet of water. And so we did this because we feel that these deeper sites are actually better for long-term survival. And even though Acropora was historically found quite shallow, it is now most of the wild corals that we're finding are on the edge of our wall in about 50 to 70 feet.
Okay, I think that's all of the questions, and we're just posting a link now to more information on the website.
Great. We're just posting a link now, as Abby just said. And we'd like to thank you again. And of course, thank our funders, RESEMBID, the European Union, and Expertise France, as well as Stuart Walker's Humphreys, the Disney Conservation Fund, Cayman Water, the All Trust, and DART. And thank you again for tuning in. And we hope you enjoyed and learned something about coral restoration.